Thank you. So our next speaker is Professor Dennis Jomes from the University of Greenwich, and he's going to talk about the 3D printing for the pharmaceutical and medical applications. Dennis, can you hear me?
Yeah, definitely. Let me share my screen.
Yes, now we can hear it. Yep, we can see it. Yeah.
Can you see my screen as well?
Yes, please.
Okay. Thank you very much. I'm the last one for this wonderful seminar. So as Adi mentioned, that I'm going to give you a quick overview of 3D printing applications for pharmaceutical and medical work that we have done in our lab. Before, let me see if it works. Yeah. So quickly, a few things about our group. As you can see on this slide, we're a multidisciplinary group. I'm currently the director of 2 research centers at the University of Greenwich, but we have a broad spectrum of areas that we are very active. So we cover biomaterials, continuous manufacturing for pharmaceutical applications, additive manufacturing, nanomedicine, and the last couple of years we started dealing on food technologies, mainly through 3D printing applications or techniques. So we heard before about 3D printed stents and the concept of developing these medical devices. Just to let you know that historically, more than 10 years ago, there was a strong interest of developing biocompatible, biodegradable stents by using different approaches, different techniques. And the idea was that it would be ideal for the patients that after a while, after a few years, the stents that are usually metal alloys or simple metals, they would be dissolved or absorbed by the body, and then the stent will disappear but the artery will remain open. So we started as well years ago in 2015, and then the concept was to develop what we call print personalized stents, or that could be either empty or loaded with a drug substance. And the concept was to use those by using 3D printing, develop those by 3D printing, because we could print complex geometries and also adjust the shape and the size. As a result, we'll be able to improve the clinical outcomes. And not only that, but this could be done by not only by tailoring the size and the shape, but also the drug release profile using specific certain specific drug substances, and at the same time the technology would benefit by reducing the material losses. What you can see here, these are some biodegradable and polymeric stents that have been made over the years, and some of them are already under clinical trials. And it was found that not all of them work because they have some critical pitfalls. For example, the high surface contact surface area with the arteries could cause a problem. All of those you can see are made by laser cut. So our idea was to use 3D printing, and the project was funded by the European Union, a program called Interreg, in 2016. So basically, in the very beginning, we wanted to use fuse deposition modeling, and the reason was that there's a very good explanation about that there's a number of medical polymers already acceptable in marketing that we could use without need any regulatory authorization. As you can see, we could design a number of stent designs, and we already know that the design of the stent is very important. We cannot. It depends on the properties of the polymer, of course, but the design itself could facilitate a better stent performance. So we started using fuse deposition modeling based also on our experience, because we could develop those materials by extruding them. You can see a video clip here. So how we extrude the material, we make the filament ourselves. This is a small extruder where we have a 3D line. We can collect the filament. This could be an empty filament, or a drug-loaded filament. So there's a lot of different options. So after the collection of the filament, which is the process is highly accurate to avoid any defects during 3D printing, we could use our polymers. Also, the idea was that we can use a combination of polymers by optimizing extrusion in order to improve mechanical properties. So before, of course, we waste a lot of material because in order to process one of these extruders, we need at least 20 grams per material, and the polymers can be quite expensive. We ran a finite elemental analysis with, in collaboration with our engineers, and we try to simulate stents in real time. So we could get a lot of data, and by evaluating the stent design that and some of the properties like radial recoil, longitudinal retraction, force sorting, and max diametral strain or max rest, would save a lot of time by only studying the mechanical properties. And once we identify the right composition or even the right design, they could go and start printing the massive amounts of these or make some of these filaments to use for 3D printing. So again, we can see some of these simulations that were done by my colleague Michael Okereke, and that save us a lot of time and, from experiments and also of material. So what we did then, we moved to 3D printing of these different designs and by using FDM. And as I mentioned before, the capability was to print plain designs and apply the drag on the surface by using another technology which is called inkjet printing. And in that case, we could control the drag release profile. So we know that it's different. The release rates are different when the drag is coated on the surface or when it's embedded within the stent matrix. So you can see some of these different designs that on the left where we could print, but of course that doesn't mean that all of them are successful. You can see in the middle image the different release profile depending on the deposition of the API on the surface. We could play a lot with that. This is a video clip which shows how we can print a stent by using FDM. So the printing time for a miniature device like that was about 2 minutes, which was quite good. So that was the first generation of stents that we started developing. However, as you can see here, we had this minor problem or major problem. The surface is not very uniform. You can see the different layers on the surface, and also the other thing was that the thickness of the stent was difficult to take below 300 micrometers or between 300 and 500. Also, we would have to apply a post-processing technique in order to make the surface smooth, because this can affect the clinical results of the stent. However, one of the things was that we managed to, by using certain combinations of these materials, we were able to make 3D printed stents by FDM, crimp them on a balloon, and then try to expand the balloon and simulate the process and see if the stent is going to withstand the crimping and the expansion. And you can see a video clip that we ran that was quite good. Further evaluation showed that the stent was quite robust and it didn't break. We didn't have any defects after the expansion in balloon angioplasty. However, as I mentioned, the major problem we had was that we couldn't, due to technology limitations, we couldn't take down the thickness of the stent below 300 micrometers. And we know from the metal stents that it has to be at least 150 or let's say around 200 micrometers. So we then moved to a different technology. Probably you're aware about that. It's called stereolithography. And then we started working on our design. We started designing our own resins by trying to use bio-based and bioresorbable materials. And the reason that we moved on SLA was that it offers several advantages. It has excellent print resolution, can go down to 25 micrometers. We could develop our resins to offer adequate mechanical strength and also fabricate what we try to do, or we call safe memory stent scaffold. So at the same time, we could incorporate drug substance during the printing process, as I mentioned before. But also we could also print plain stents and apply the drug on the surface. Eventually, as you can see on this image, on the top image, we could print a wide range of different shapes, designs, or dimensions of the stent. But what was important for us was that the thickness of the strut could go below 300 micrometers. We could have easily 150 micrometers or 200 or 250. And then another benefit was that the surface was very smooth, and we didn't have to do any post-processing work. So we ran some microtomography with some colleagues from Southampton University, and as you can see here, we found that gave us a good idea about the printing process. There were no defects or cracks on the surface. It was almost perfect. We also ran and very consistent. This is very important. We also ran some degradation studies, and we saw that for the resins we design ourselves, or we try to develop, there was a degradation about 12.5% over 3 months, and this was also really good because those stents are supposed to be mechanically strong for at least 24 or around 24 months before they start degrading completely from the body. And also by the time will be covered, they will be fully covered by endothelial cells, so there's no damage on there's no cracking or breakage of the stent that will go into the blood circulation. So the next area of interest was again using stereolithography, was to use some resins that we develop in the lab, or resins that we can buy, dental resin grades that we bought off the shelf from the companies, to use it for 3D print of microneedles. For those that are not familiar with microneedles, these are miniature devices that can be used for drug delivery through skin. They can pierce the skin without causing any pain or reaching any nerves on our body, and then they can deliver the drug substances, either those on a solid form or as a liquid formulation, as we'll see later on. The benefit of using stereolithography again is that we have an excellent resin biocompatibility. Those dental resins have been approved by regulatory authorities. Of course, we have a very high resolution for complex designs, as we can see later on, and high mechanical strength. That was very important because then... Yes? Hello?
Please unmute yourself, Dennis. Dennis, you are on mute. Dennis, can you hear me?
Now?
Yes, I can.
Okay, I don't know what really happened. Let me go again. So what we can see here, I don't know if you heard about microneedles when I started talking about microneedles. Did you? Is that okay? Should I continue from here?
Yes, please.
Okay, so those microneedles here, you can see that on the top image we have empty microneedles. The bottom images we code the active formulation on the surface, and by using this inkjet printing. On the right image, you can see that this is a inkjet printer, and we can design the algorithm to coat the material on the surface without any losses or any satellite material going onto the substrate. What we did, of course, we evaluate the pressing capacity of these polymeric microneedles, and we found that the shape of the microneedle is very important. Depending on the shape, we can apply different forces to pierce the skin. This was quite critical, and we found that the piercing forces are less compared to metallic microneedles. Also, we ran some trials to see how long does it take for the skin to absorb the material and also recover, so we don't have any holes on the surface. In about 4 hours, we see that the holes are closing on the skin. This is perfect. We ran also some optical coherence optical coherence tomography, and we found that depending on the aspect ratio of the needles, the penetration depth can go up to 82%, which is also perfect for our work. Furthermore, we ran some micro tomography analysis, basically to investigate if those micro needles have any defects, if the printing, the body of the needles is uniform and we don't have any internal defects. We didn't want them to break and remain in the skin. And at the bottom image, you can see that we ran some work. It's not pretty clear, but we want to see if the coating is quite adhesive. So there were worries that the coating, when we apply the coating, it might be removed from the surface of the needle. But we found that by using optical coherence tomography, that the coating is quite adhesive and it doesn't leave the surface, but only when it hydrates. So these solution profiles, we found that in this case we wanted, one of the first work we have done with actually Dimitris Lambru was to apply insulin on the surface and use these micro needles to deliver faster, in a rapid way, insulin, which is it's a large molecule. However, we find that by using France cells, the in vitro dissolution was quite rapid. Within 10 minutes, we had more than 80%, between 70 and 80% of insulin delivered in the receiving compartment, which was really good. Of course, we all know that this kind of in vitro evaluation is not always indicative of what's going to happen in vivo. And for that reason, we ran some animal trials. And we compare that with subcutaneous injection to see what is the effect of microneedles and if they offer any advantage by delivering insulin in vivo. So what we found was that, again, I'm not going to go in detail, but we found that the duration of plasma insulin by using microneedles was much better. It would lasted at least for 4 hours, and the same thing with the glucose levels were reduced for longer period in the blood plasma, but also the insulin levels were much higher. But when we use microneedle administrated work, microneedle administrated insulin. So we prove that the clinical benefit of microneedles is obvious, and also we run some more trials, not only with large molecules like insulin, but also small small molecules for cancer treatment. I'm not going to go in detail, but we prove that for skin cancer, 3D printing microneedles were successful to deliver small molecules like curcumin or paclitaxel and cisplatin. So this was an area we expanded our research since 2016 on 3D printing microneedles, because another major advantage is that they offer, by using 3D printing, it's very easy to scale up the technology. At the moment, there are no products in the market on microneedles because the scalability of metal microneedles is very difficult. And another work that we have done was to, of course, I mentioned at the beginning that the material can be coated on the surface, but also it can be administered as a liquid. So we developed what we call hollow microneedles by using stereolithography, and that those microneedles are hollow, and then they're interconnected through microfluidic channels. So and on the side we have a small hole that when we pierce the skin, we can administrate the material. So this could make our life easier, especially for formulations that we need to act very, very fast. And the prototype was developed through one of our PhD students we had with Dimitris. And as you can see here, we could administrate the liquid formulation by using a diaphragmatic microdosing pump, where we can control the amount down to 5 microliters. There's a video clip here. We can see that by using a small number of needles on the array, it is very easy to administrate large amounts and very fast. of liquid formulation. So you can imagine for humans, you just need a couple of these needles per array. This is an optical microtomography study, optical coherence tomography study we did with colleagues from University of Kent, and we actually wanted to see what really happens to the liquid when it goes under the skin. That was quite tricky, and what you see, the liquid we'll see here is we use milk because a lot of other liquid formulations didn't work. And what we found, this is another video clip, we found that the liquid goes under the skin, creates a depot, and then soon it disappears. So it is readily absorbed under the skin. And that was really good to show us that we can have a very quick effect when we administrate any liquid formulation. I'm not going to show that, but we also ran some animal trials, and with the results we found were quite robust and very interesting. So the last bit of my presentation is it has to do mainly on pharmaceutical applications. This is a work that we did with, the work was funded by Bristol Medical Squibb. And basically the idea was to identify technology that can be used at the point of care. So the concept was to develop a technology in a way that we can install it in a hospital or in the future in pharmacies, and then we can use it to print different formulations. So for this person, we use a technology called micro extrusion. So basically, we use a bioprinter, which we turn that from cell-ink, and we turn that to a printer that we can print. We can print thermoplastic materials or polymers with blend with drug formulations. So I'm not going to go in much detail because we don't have much time, but there are several benefits of using this technology. The most important benefit is that we can use the formulation in a cartridge, so the clinicians do not have to worry about pre-processing steps. The formulation will come in a cartridge, they just plug it in, and then they can start printing the dosage form. And of course, the beauty of this process that has a small footprint and it avoids any cross-contamination within the hospital. So then the next step was to decide which formulas we are going to print. There's a lot of work in around European Union discussion about 3D printing the medicine for children. They offer several advantages, but the most important is that they help to increase the adherence of children because they don't adhere to their medication. And the reason is that we can increase palatability through taste masking improvement of those formulations. Just for your interest, we can see here that we could print, using this technology, we could print a range of formulations. We can play with the infill density of those materials, and that means that we can also control the release profile of the API. We could make them chewable, so kids can chew them, and they don't create any nasty or bitterness taste. And you can see here that we can play also with the design. We can have 100% infill density of the formulations of shape that they look like confectionaries, and children think that this is a sweet that they can take. Of course, we offer, as I mentioned before, we can offer excellent taste masking by processing the API with the polymer, so we force them to interact. But at the same time, we can add some sweeteners or flavors, and we increase the palatability of those materials. We also run some in vivo trials by using panelists to evaluate the sweetness of the 3D printing formulations and the aroma. And the beauty is that we can use a wide range of flavors like strawberry, banana, or orange, depending on the taste or the perception of the children. And on the side, you can see some dissolution profiles that actually, for those who are not familiar, show that the release is almost immediate. Within 10 minutes, we have a release of the API, which means we can have a very fast onset action when this is administered to children or any patient. So these are some of the benefits. So in conclusion, 3D technologies offer the ability to develop personalized medical device, but also pharmaceutical dosage forms. We can control the geometry, the drug dose, the release in all these kind of printable devices or dosage forms. And of course, we know that 3D printing can overcome some pitfalls or limitations of existing technologies and improve the clinical performance of stents or even other medical devices. Of course, we are there are more improvements in the technology. We know that over the years the technologies improve. We get 3D prints that are much better, have better resolution or better features, or even new materials. So, as I usually say, we are in the beginning of this 3D printing era. There are more to come in the near future. I would like to thank my group. Most of these guys are not in the group anymore. They moved to academia or companies. And also I would like you for your attention. Happy to take a few questions. Thank you very much.
Thank you so much, Dennis, for your great presentation. Any questions, comments from the audience? Actually, there is one question. Let me. Yeah, so Yamis has a question. How could you include drugs during the extrusion of the filaments? And will these drugs withstand the heat of the, deposition or? Yeah.
Correct. This is a very good question. Yes, the first thing we run always when we use extrusion, we're going to run some thermal analysis, thermogravimetric analysis to see if the drug is sensitive to heat. So usually we select drugs that they are not very sensitive to heat. This is important. If we see that degrades, we cannot use them. The way to incorporate during extrusion, there are basically two ways. One is you make a physical blend with the polymers and the APIs, and then you can process it. Usually those medical grade of polymers, we use resomers that the extrusion temperature is not very high. However, some of them, when we wanted to make a blend, they don't come as a powder. And then the API is always in the form of powder, so the blend is not going to be uniform. So this is where the benefit of extrusion comes. You can start extruding your blend of polymers in the form of pellets or even as granular material, and then at a different stage you can add your API and blend it while the polymer is melted. You just need to have a second feeder for the API. So that's how you blend both of them, and then the extrusion ensures that the blending or the distribution of the API within the polymer matrix is perfect. It's very homogeneous.
Brilliant, thank you. And the second part of that question is regarding the ceramics. Do you use ceramics as a part of the drug printing?
Sorry, I lost that. I couldn't hear.
Can you hear me now?
Use
ceramics. Do you also use the ceramics to print the drugs? No,
we don't use ceramics. It has to be a polymer which is biodegradable, 100%. So again, even lithography, we either incorporate the API in the resin, and that depends. In some cases, the API dissolves the resin. If we cannot do that, then we print empty stents, and then we apply inkjet printing on the surface. We have developed a pattern to apply the coating on the surface of the stent.
Okay.
Ceramics could be used, but they have to be biodegradable.
Thank you. Any other questions? No questions from the audience?
Thank you very much.
Okay, so yeah, there's no question. Thank you so much.